Hello, Mr. Ullman here, and this is your Ullmancast on the eye. One of the fundamental characteristics of life is that all living things are capable of responding to stimuli in their environment. Now, in order to be able to respond to what's going on around you, you first, as a living thing, need to be able to sense what that stimuli is. Now, we have chemoreceptors on our skin and on our tongue. We have physical receptors, mostly on our skin and scattered throughout the body. But if you've ever walked around blindfolded and felt that disoriented feeling, I'm sure you understand and would empathize with the point of view that our sense of sight is the one that is the most important. This video is going to focus on how it is that the eye perceives and then passes on that information to the brain. First, let's review the sense. A sensation is going to start with a stimulus. A stimulus needs to be received, and the cell or body part that is going to receive that is known as a sensory receptor. That receptor is going to translate that stimulus into an action potential, and that action potential will then be sent to and interpreted by the central nervous system in the brain. The type of receptor that's capable of perceiving visual stimulation are called photoreceptors. Photo meaning light, and receptor being the type of nervous cell that's actually receiving that information. They're going to respond to electromagnetic radiation. And the size of the wave is going to determine whether or not it's going to be perceived by the living thing. Now, UV light and visible light are the ones that are going to be emitted primarily by the sun, and so therefore those are the things that are bathing the earth in energy on a daily basis. As humans, we're not capable of perceiving this ultraviolet light, and I'll touch on a little bit about how that's different in other species. If we look at, in the very simplest form, an eye spot. So they have structures on them that have a simple set of photoreceptors which will allow those organisms to perceive the presence of light. And they will then be able to exhibit a type of behavior known as taxis that will either draw them towards or away from it. So if they, like the planaria, if they want to stay in dark, murky areas in order to ensure that they can be hidden from predators, their eye spots, when they perceive light, will lead them to move away. And so that's called negative phototaxis because they're moving away from the stimulus. Something like a worm is also going to have that same type of behavior. As you look at arthropods have compound eyes. And a compound eye just means that the eye is broken up into individual parts. So each one of those little structures present on the surface of the eye does act independently as an eye. It has its own cornea, it has its own photoreceptors, which are then going to transmit information in the form of action potentials onto the brain. Now, arthropods bring up a good example in that as we look at the animal kingdom and the types of photoreceptors that are present, not all photoreceptors are the same. And so while in humans we see that the photoreceptors allow us to see only what's in the visible light spectrum, there are a number of species that are capable of seeing wavelengths of light in the UV spectrum. And that ultraviolet light, which does come from the sun, may reflect off of things like the flower present here, and even though to us we can't perceive that particular form of light, to an animal like a pollinator, they may be able to see that reflected UV light, and then that's going to be additional visual stimulation that's going to draw the pollinator towards it. And so that would be a nice evolutionary relationship showing coevolution. Because if there is a spectrum of light that most organisms are not capable of seeing, but the pollinator is capable of seeing it, then it's to the advantage to color for the flowers to be colored in a form of light that is going to be specific to its pollinator. Now, while that's true for UV light, that would be just as true for a blue or a red or an orange or a yellow flower. The color that the flower is, meaning the type of light that it's reflecting, is certainly going to be a type of light that will match up with the photoreceptor present in the eye of its pollinator. We need to spend most of our time, however, on the vertebrate eye. And so this basic eye structure is going to be seen in all vertebrates, meaning that reptiles, amphibians, mammals, birds, we're going to see overlap in terms of the parts. Obviously, there's going to be some differences in terms of particular shapes and relative sizes of structures, but the basic body plan here of the eye is going to be in common between all of these things. So as we look at the diagram, obviously you do need to be able to identify each of these parts. So I'll give you an opportunity to do that in a moment, but right now I want to review some of the functions as you're looking at some of these different parts. Starting from the outside of the eye, we have the cornea. The cornea is on the outermost part of the eye. It's transparent, so light will pass through it. It is flexible, which ensures that if anything bumps into the eye, it's not going to be brittle and shatter. And it's fairly tough, so it is going to provide some protection for the more delicate parts of the eye which are underneath it. What's interesting about the cornea is that this tissue actually doesn't rely upon blood in order for oxygenation to occur, because the presence of blood vessels would actually block out some of the light that needs to pass through. So the cornea, because it is always exposed to the air, is going to allow oxygen to diffuse in from the environment. Therefore, immediately underneath the cornea is going to be the aqueous humor, which is going to be a very water-like substance, which is going to, again, provide both some structural support as well as cushioning there. Beneath that, the pupil is actually an open area. So the pupil is not really a physical structure, but it's more a lack of a structure. We see that above and below the pupil is the iris. And the iris are a set of smooth muscles that control the size of the pupil. And so as those muscles contract, they get smaller. And as they get smaller, that's going to increase the size of the pupil. And then as they relax, they're going to decrease the size of that pupil, and that's going to be necessary to control for the amount of light that gets passed in. Because as we'll see in a few minutes, light as it passes into the eye is going to hit the retina, which is going to excite proteins that are present in cells there. And so if there's too much light, those cells will get overexcited, and you're going to lose the ability to be able to distinguish objects from each other. You're going to see everything as light. And so if you've ever had that moment of extremely bright light flashing in your eyes, you have difficulty distinguishing one object from another, and it takes some time for your eyes to be able to reset from that process. So the iris working to control the size of the pupil is important in being able to protect you from being overstimulated in very bright light, but to allow the light to come in when you're in those dim situations. Beneath, just past the pupil, is the lens. Now the lens is extremely important because this is what is helping to bend the light that comes in. As light enters into the pupil, it's actually going to be diffracted. So it's going to bend away towards all of the different parts of the retina, because the retina covers the entire back portion of the eye. And so in order to make your vision more precise, so that you can see not blurred images but very sharp images, it's necessary to bend that light and spread out the light that's entering into the eye, so that you'll be able to see some of those more fine details. The lens does change shape, so there's smooth muscle attached to it on either end, and so the changing in shape of the lens is going to allow you to focus on objects that are closer or further away. So by changing the degree to which the light is bent, that's going to adjust that focus for you. Then in the center of the eye is the vitreous humor, and this is a gel-like substance that's going to fill up the majority of the space within the eye. So the eye is mostly empty space. That provides some pressure to ensure that the eye stays fully inflated, to keep the lens and the iris and the cornea all pushing up against the front part of the eye. In the back of the eye, you have the retina. So the retina is going to be covering the surface of the eye, and there's a tough layer, several tough layers of tissue surrounding that. But on the retina is going to be the photoreceptors. So this is where light is actually perceived. The front part of the eye is really just there in order to be able to adjust the light that is entering in. The retina is really what is responding to that light. You have a part in the center of the back of the eye directly across from the pupil called the fovea. And the fovea is going to be what allows you to focus on and see very clearly what is directly in front of you. So whenever you direct your eye towards an object, what you're doing is the eye physically turns so that the center of the eye, or so that most of the light comes in, will hit the fovea, and there's going to be a greater concentration of photoreceptors here. That'll be important in terms of color perception, which we'll get to with rods and cones in a minute. You do have one part of the eye where the optic nerve is located, and that's actually a disruption in your field of view. So at that optic nerve, you actually have a blind spot because there are no photoreceptors on that point where all of the individual neurons where the axons are coming together to form that optic nerve. So now that you've seen some of those different parts of the eye, take a moment now, test your knowledge there, see if you can identify the parts that are here. Obviously, if you have a little trouble with that, then go back and review the previous slide, come back and try it again. Alright, so what is happening to light as it comes in? So light is focused by the lens onto the retina, as I said before, and the retina is going to be the lining across the back of the eyeball. We'll get into the structure of the retina here in a moment. The retina is going to be where the light energy is going to be converted into action potentials, which are going to be sent to the brain. Now, one thing that's interesting is that we have something called contralateral processing. Now, you're probably aware that for example, muscle control works such that the left hemisphere controls the right side muscles on the right side of the body and vice versa. Now we have a similar case here, but it's not what you would expect. It's not that the left eye is information is sent to the right part of the brain and vice versa. It's actually a little bit more complicated. Things in your right field of vision are anything in your right field of vision is going to be sent to your left hemisphere, and anything in your left field of vision is going to be sent to your right hemisphere. In other words, if you took your the center of your face and divided that, made a line going straight forward, everything to the right side of you, no matter if you saw it with your right eye or left eye, that information would be sent to the left part of your brain. And anything that you see to the left of the center is going to be sent to the right part of your brain. All right, now starting to look a little more at the retina and the photoreceptors. Rods and cones are going to be the two types of photoreceptors. Rods interpret presence or absence of light, so just there is light or there is no light. Cones are going to respond to different wavelengths of light. If we look at specifically what's in there, a rod cell, which is pictured here on the top, is going to have a series of discs in the top, which are going to have photoreceptors in them. That's going to then communicate through a synaptic terminal with an additional cell that it will be attached to as part of the nervous system. Cones, they're called cones because literally you see the difference in shape here between what's on the end of those cells. A different set of discs with a different set of photoreceptors on them. Within those discs that you see in both of those photoreceptors are going to be proteins. The most important protein that you should know as an example of a photoreceptor is called rhodopsin. And rhodopsin is going to be a protein that will change shape, that will physically change shape when exposed to a particular wavelength of light, depending again on what type of protein it is. So those proteins change shape when responding to light. And so what happens is that when light occurs, the protein changes shape, it opens up sodium channels, which causes a hyperpolarization of the cell. That's going to prevent glutamate from being released, which is then going to hyperpolarize or depolarize, depending on the type of receptors that it has, the bipolar cell that it's attached to. In terms of response to dark, rhodopsin is inactive. The sodium channels are open, which is going to depolarize the cell, and depolarization is going to lead to the release of glutamate, which in this case is the neurotransmitter. That neurotransmitter will either depolarize or hyperpolarize the adjacent cell. Looking more specifically at the way that the retina is wired to your optic nerve. So we have a set of epithelial cells at the top, so this is the back of the retina, and you notice that light is coming in here from the front. So you can imagine that the front of the eye is going to be at the bottom of your screen. So light is actually going to pass over the nerve and the initial neurons to be received by those rods and cone cells, which are attached to the back of the eye. So those photoreceptors are there, and one thing that I want you to notice is how it is that they're wired. So here the blue cells, the blue rod cells, there are several of those attached to the next layer of cells, which are called bipolar neurons. So you can see that in this particular picture, about 3 rod cells, it actually could be more, but multiple rod cells are attached to a bipolar neuron. That bipolar neuron is then attached to a ganglion cell, and that ganglion cell is then going to send an action potential off to the brain. So if we do a quick comparison here between these two different types, rod cells are going to be dominant in dim, whereas cone cells are going to be inactive when it's dim, which is why you can't see color when the light is very low. There are 3 types of cone cells in humans, whereas only there's 1 type of rod. Rod cells are going to be evenly spread throughout your eye. And so in dim conditions, your peripheral vision and the area that you're focusing on are going to have about the same resolution. Your peripheral vision isn't much worse than the thing that you're staring directly at. Whereas your cone cells are going to be concentrated near the fovea, which is in the center of your retina. And so your cone cell, because they're concentrated in that point, when it's very light, and your cone cells are the things that are doing most of the perception of light, under bright conditions, you're going to have very sharp vision on the thing that you are specifically looking at, and your peripheral vision is going to be a little fuzzier because you don't have as many cone cells on the outside of your retina. There is the receptor to ganglion ratio, which is important. So there are many rod cells to ganglion, which is why the acuity of these is very poor, whereas there's a low ratio of receptors to ganglions for cones, and that's why their ability to perceive things like edges is going to be very high. And with that, we'll end this video. I will leave the link to the Prezi in the space below, and remember, take care of your eyes.